Okay, good morning, everyone. Good morning. How should not be? Okay. Hi, everyone. Today I'm going to present the chapter 9. It's about an enzyme rhodopsin. It's including several different kinetics like histidine kinase rhodopsin, rhodopsin guanine cyclase, and rhodopsin phosphodiesterase. So all of these enzymatic rhodopsins actually are quite close to each other. They form a subgroup um, in the family of the rhodopsin, and especially bacterial rhodopsin. Um, but it's quite different from other like channel rhodopsin and other pump like chloride pump and potassium pump. But some very critical area like all-translating binding rod pocket that's still quite conserved. So now let's move on to see the. So today I'm going to present the enzymatic rhodopsin. So the first I show the outline. It's including the histidine kinetics, rhodopsin, and rhodopsin guanine cyclase, and the rhodopsin phosphodiesterase. And we can see here it shows that they share quite a lot of similarities, and we will discuss this later. That of these three enzymatic rhodopsin. So like one thing is that in just this structure feature is that the 7 transmembrane segments that are quite similar and share with other like pump or channel rhodopsins are quite similar, but that involves a transmembrane numbered with 0. It's a very unique feature for all enzyme rhodopsins known so far. So we already know that other pumps, their N-terminal are extracellular and the C-terminal are intracellular. However, for this, with an transmembrane 0, so adding a new transmembrane, so it's like those rhodopsins, they go transmembrane one more time, and this makes that their N-terminals and the C-terminals are both located in the cytoplasmic site. And all of these three enzymes forms a dimer. So the functional like dimer. So I think for like a kinetics, the histidine kinetics probably is quite critical for them to like interaction. However, other dimerites probably is due to the property of those transmembrane domain of those binding with the all-trans retina. And here they forms like by naturally they form a dimer, just like other pumps or channel rhodopsin. So I mentioned before that this enzymatic rhodopsins, they forms a family that are slightly different with the pump and is very little even more far away with the channels. However, the all-translating binding pocket is quite conserved between like channel rhodopsin and other bacterial rhodopsin, and with this enzymatic rhodopsins we can see here, it's quite conserved. And the critical part for iron selection, it shows a different among the two different histidine kinetics, and also comparing to other pump or channels. So first, let's focus on histidine kinase rhodopsin. So this is first discovered also in a green algae, the Camerotic Rhodopsin adopsin. So it shows the sequence have a several functional compartments. So we can see here that's a quite a familiar creature, and that's the green algae. So it, of course, it has the transmembrane and also the photosensor center, and also another intracellular part that histidine kinetics, and the response regulator RR, and the guanine cyclase domain, the GC part. So the histidine 352 in HK domain was identified as a phosphorylation site, and that's critical for the function of this kinetics. The phosphorylation transferred to D1092 in RR domain in the response regulator. Now let's take a look at some information about rhodopsin guanine cyclase. So this rhodopsin guanine cyclase, there's some critical site. For example, if you change like this two site, like induce a double mutation, it will convert and this substrate to the GTP to ATP. And these mutations will also result in increasing dark activity. Partial truncate of N-terminal domain result in high dark activity and low light-induced second GMP production. So actually, this transmembrane zero and linked with the N-terminal is quite important for the function of this light modulation and activities. So that's light regulation of enzymatic activities. And then this RhGC shows high activity in presence of Mn lalum in the magnesium, which shows you observe type 3 guanine cyclase. Cyclase receptor specific is defined only by two residues, like ENC guanine cyclase and KND for adenine cyclase at these two positions. Let me check. Yes, it's the same positions. So this the two critical point site determine the binding site, determine which the substrate they was using. And this is quite different from the PDE, the rhodopsin phosphodiesterase. There's no very subtract like selection either to change any site, to change the preference to like cyclin GMP or cyclin AMP. So that's quite different between this guanine cyclase and phosphodiesterase. So here we can see that more clear diagram about the transmembrane function. And we can see here, comparing to like channel rhodopsins, so there's a very large N-terminal parts, and that's critical for the enzymatic modulation by the light. Of course, a C-terminal because of the regular site, and there's a linker site, and the kinetics. The guanine cyclase is the enzyme part of the rhodopsin guanine cyclase at the C-terminal. And this image indicates that the dark adapted state have a maximum option at the green light 525, and this is quite a long wavelength, and this is so far known by nature that is the longest wavelength of the stimulation. And in this paper, they try to link the YFP at both the N side and the C side, and actually it's truncated that they removed the enzyme part of this enzyme and only keep the N-terminal transmembrane part and a little bit C-terminal like the linker, and then remove the enzymatic part. For they also in the same structure that's like they call it CAGC, and it's a market-long photocycle in which the NM intermediate lasts much longer than the Y-type version. So in another paper, people think that a beta sheet forming in the linker part is very critical for the modulation and the function of this guanine light-gated regulated adenine cyclase. And they call it like a TOM. It's a 2 beta sheet that is coming from like this structure, but inserted further into the guanine cyclase enzyme part. So it's a tongue-like extrusion, consider hairpin that recovered into the AC subunit and is sandwiched between the helix bundle of it and adenine cyclase domain core of other chains. So let's see that this beta sheet forms really different locations in the dark state or under the light, and then this modulation will change the light dependent enzymatic functions of this guanine cyclase. Now let's move on to see the rhodopsin phosphodiesterase. So rhodopsin phosphodiesterase is found in this flagellate. And first time when I see this creature, I find it's so similar to the photoreceptors of the mammalian retina. So before Darwin have said that and although he created this theory of evolution, however, he mentioned that just look at the anatomy structure of the retina and see that the design is so delicate. It's very hard to think about it like it's be by evolution that involved such a delicate structure of the retina. However, I think that it's possibly that the available natural phenomenon is still not enough for Darwin at that time period. I think if he see a lot of like a microbian, and we can see that it is colony flagellate, this structure is so delicate and are highly functionized with a polarized structure. And it can also forms like a group of cells that stick together and with some kind of orders. And I think that is the basic part of like our retinas, how to develop upon of these variable options or parts. So in 2017, and these two group, they both independently find this novel type of enzyme rhodopsins, and that's rhodopsin phosphodiesterase. It's an N-terminal cytoplasma domain which is made up of a 60 amino acid. It's N-terminal. And additional, and it's very interesting part that for other like rhodopsins or for other rhodopsins, it's not reported that a fluorescent protein located at N-terminal, C-terminal will dramatically affect the function of the target protein. However, for these Rh-PDE proteins, located at the C-terminal will probably hamper the conformation change for enzymatic activities. So avoid like link those fluorescent protein at the C-terminal, and placing YP at the N-terminal will result in improvement of light activities. And this is highlighted by this paper. And then we can see that the structure of this phosphodiesterase. So you can see that as a GAF part A, GF part B, and the enzymatic part. So in the dark, the PDE is inhibit constitution activity towards the cyclin GMP or cyclin AMP. So in the dark, so it's continuously like phosphodiesterase like cyclin AMP or cyclin GMP. And the light will stop this function or like inhibit this function to make this protein to an M state. And after 4 seconds, and the state will be changed to original state, and then go back to the constitutive activity that continues phosphodiesterase, cyclin GMP or cyclin AMP. And this study is working on mammalian set, that's a HEK cell or the frog oocyte. So a pretty new paper, and that it find that this group is from Berkeley, and I go to the European and Caribbean and sea area, and I find I discovered several like this rhodopsin phosphodiesterase proteins, and I discover the functions on this coinoflagellate. And it's so interesting that it seems that in the dark, that they prefer like to like from like a structure, this group like tend to like the flagella like inside the society, and after shedding the light, and they will change their they change the whole morphology of the group, this colony, and they change it. So and the refrigerate will towards the outside. So this is a this light change of function is considered to be like light dependent manner, and it's related to the intracellular cyclin GMP level. And this cyclin GMP signal cascade might trigger atomine-mediated apical contractivities. So it's I think it's quite interesting, and it should be highlighted in this lecture. Because it directly linked to the light regulation of the behavior of a group of those creatures. And like goes to another group, a Japanese group, and also the author of this paper, of this chapter, and they discover that they compared many different Enzymerhodopsins, and I find that the Rh-PDE1 its activity majorly is the cyclin GMP, and 4. So they do this study on HEK cells. So some the PDE2, PDE3 is not functional in HEK cells, and the PDE4 is also light responsive and active towards cyclin AMP. And we can see again here that E is towards cyclin AMP, and first is cyclin GMP, first is cyclin GMP, and the fourth is said that it's towards cyclin AMP, but the function is quite different. It's not so different between light and the dark and the light state. And the most stable, like just show no difference. So with light or without light, so this is Rh-PDE is continuously have a very high enzymatic function. So I still think that like the PDE1 is quite like be a candidate for the light regulation of the function of the cellular functions. So this Y type, and we see that after stimulation and cycling AMP, enzyme adenine saccharase and the cyclin AMP's concentration is continuously increase, both under with the control, negative control, or a mutant control, and the PDE-Y type. After shedding the light with the 510 nanometer, and there's a decrease in cyclin AMP level. So to zoom in, we can see here as a dramatic difference between these Y types. So mean that at this part that the light can start to stimulate, like shows a decrease in cyclin AMP as a decrease. So and this is quite repeatable, like addition of rhodopsin to an adding another saccharase, and this is a concentration increase. And after stimulation with light, and the Y type 1 and can repeatedly like reduce the concentration of the cyclin AMP, and it is quite repeatable and is reliable. At the last part, we're going to compare the light-dark ratio of these three different enzymatic rhodopsins. So, the ratio of the HKR is about 35 and the PD less than 5. So not much quite different between light and dark stimulation. But the rhodopsin guanine cyclase is a quite different, that the activity is quite dependent on the light. And this is a reference, reference that is being cited in this lecture. And thank you, everyone. Thank you.